A very good afternoon to everyone. Welcome in the class. The topic of today's class is patch clamp technique. And as we say in the beginning, let's crack CSIR UGC NET together. And that is the name of our channel also. Let's crack CSIR UGC NET. A very good afternoon to everyone. Welcome. And we are doing the crash course for GATE 2021. So in that we are going to talk about patch clamp technique. I'm your educator Farida Bandookwala. I have teaching experience of 10 years. I've qualified MP SLET examination in year 2018. You're opting for Unacademy. What all things you are going to get? You're going to get daily live classes so that you will be into that momentum of studying all the time. And in live classes, you can ask your doubts and queries directly with the faculty members. The whole syllabus is quite structured so that you can complete all the 13 units or the syllabus of GATE in a predefined time, because to complete the whole syllabus in a systematic manner so that you can cover all those topics which are actually important according to exam point of view is quite important. Live tests and quizzes are taken so that you can evaluate yourself that how much you have learned so far, and whichever category you are choosing for, then you will have unlimited access for that particular category. In free live classes, you are going to get the free live classes on the platform of YouTube, that is on the channel Let's Crack CSIR UGC NET. You just need to subscribe the channel. For special classes which are again free classes, you just need to download the Unacademy learning app. By downloading this app, you will be asked to make your account. So make your account and use my referral code FB01. By using my referral code FB01, you will be able to attend my special classes, my free special classes, and the special classes of other educators. Recently, a discount is going on in the Plus subscription. And what is this Plus subscription? This Plus subscription is a paid subscription, and all the 13 units of CSIR and all the units of GATE are covered in this Plus platform. Like in special classes and YouTube, we try to cover maximum things, but in Plus, each and everything would be present in a well systematic manner. All the 13 units are you can access them through the Plus platform, and you need to pay a certain amount for this. And as I told you, recently a discount is going on, so for 12 months it is going to cost you 11,550 rupees, and for 24 months it is going to cost you 16,800 rupees. Additional 10% discount can be availed by using my referral code FB01. So do not forget to use my referral code FB01. After using my referral code, it is going to cost you 10,395, and for 24 months it is going to cost you 15,120 rupees. So this was a little introduction about Unacademy and the courses. After that, coming to the topic of today, that is patch clamp technique. Patch clamp technique. This patch clamp technique is basically a electrobiological technique. It's a electrobiological technique. This technique, electrobiological technique, is used for measuring the electron or membrane potential. Used for measuring electron or membrane potential. Membrane potential across the neuron. Across the neuron. So mostly this patch clamp technique is used for neurobiological issues, but for other things also it can be used, but mostly used for neurobiological issues. So if we are saying that it is used for neurobiological issues, that means it is used to understand the transmission between those cells, between the neurons, that how the neurons, they release the neurotransmitter and how they reach the other cells. Those whole sequences can be understood through patch clamp technique. So it's a laboratory technique. It is belonging to electrobiological science or electrophysiology science, and it allows you to study single or multiple ion channels in a cell. Allows study of single or multiple ion channels in cells. So this patch clamp technique was actually given by two scientists. The name is Sackman and Neher. So developed by Sackman and Neher. And this was in the year 1970s and early 1980s. So Sackman and Neher, they developed the technique of patch clamp method, and they received Nobel Prize for the same in the year 1901. Received Nobel in year 1991. So there are some scientists who have some contribution towards the patch clamp technique. So historical development. I'll be writing the name of the scientist and I'll be telling to you the things those scientists have done. So Jain, Swamendram, he did the earliest experiments in electrophysiology. After that, Luigi Galvani did the first uh, experimental evidence of electrical activity in animals by using metal wires in the frog muscles. After that, next scientist, that is Hawking and Huxley, they both gave the first intracellular measurement of the action potential in the giant squid axons. After that, Graham was successful in implanting the micropipettes and micropipettes developed by skeletal muscle fibers. After that, Cole and Marmont, they gave the voltage clamp technique combined with the micropipettes. And finally, Sackman and Neher, they gave the patch clamp technique. So this is a historical development of patch clamp technique. So here, before Sackman and Neher, there are certain people who were doing the line of experiments in the same direction. Now coming to the need of patch clamp technique. Need of patch clamp technique. So first of all, patch clamp technique is a refinement of voltage clamp technique. Right, it is a refinement of voltage clamp technique. Then it provides for low noise recording of current. I'm going to tell you the whole thing also, but before moving to that, let us talk about the need. Provides low noise recordings of current. Provides low noise recordings of current. Provides access to the inside of the cell. Provide access to the inside of the cell. By this technique, we can insert an electrode into the cells. We can insert electrode into cell. By this method, patch clamp technique, we can even change the intracellular fluid Then it creates a seal which is impermeable to iron flow. High electrical resistance creates a seal impermeable to iron flow. Creates a seal impermeable to iron flow. High electrical resistance. Patch clamp technique allows one to measure the current through ion channels versus voltage, time and temperature. Measure current, measure current through ion channels versus voltage, time, temperature. So these are some of the need of patch clamp technique, and it was given by Sackman and Neher. Now we'll be talking about the basic principle of this technique. So the principle of the method is to isolate a patch of membrane electrically from the external solution and to record the current flowing into the patch. So that is the method. But if we are taking a cell and cutting a small part of the membrane, whichever channel we want to study, and if we are just cutting that part, so the whole cell would become dead and there will be no exchange of material when it's dead. So the principle behind the use of patch clamp technique is to use a alive cell. If the cells are not alive, it cannot be used. So in the basic, we want to study just one ion channel, but we cannot separate this ion channel from the whole cell. This is the most important thing. Now, how is this achieved? That is the main thing. That how would it be achieved? So this thing we are going to talk in this. So first of all, the principle of method is to isolate a patch of membrane electrically from the external solution and to record the current flowing into the patch. Like see, if this is the cell and there are various ion channels for sodium, for chloride, for potassium, and we just want to study one, that is for sodium channel. So if we are cutting the membrane out, it is going to be very difficult. It is not going to be difficult, but it is going to lose the essence of the whole process. So what we can do is we can fix a pipette over here so that the whole thing is change. There are number of channels in the neurons, and these number of channels in neurons are always greater than the number of channels in the normal cells. So now the main task is to hold this cell at a certain place. So this is achieved by pressing a fire polished gas pipette which has been filled with suitable electrolyte solution against the surface of a cell and applying a light suction. See, this is the cell and there are various channels. There are various channels. So now what we'll do, we'll just press a fire polished glass pipette against the channel which we want to study, and this will be filled with the electrolyte solution so that it can mimic the extracellular environment of the cell. So that this is the intracellular environment for this channel and this is the extracellular environment for this particular channel, right? And the light suction would also be there so that it is holding in that particular place. So this is going to happen. So role of channel is to periodically open and close so that ions can go inside and can come out. This is the role of the channel. So that can be achieved only by maintaining a normal environment which it can in which it can exist in its normal. So all the cells, they try to maintain a membrane potential, and we call it resting membrane potential. Resting membrane potential. All the cells in the normal resting state, they maintain this resting membrane potential. For normal cells, the resting membrane potential is minus 90 millivolts, and for neurons, this resting membrane potential is minus 70 millivolts. So membrane has voltage, and that is the base level. This we call it as resting membrane potential. But for neurons, this potential is altered due to the current flow. It is kept on altering because of the flow of current. So current flow is flowing of sodium, potassium and chloride across the membrane, and they alter the membrane potential. That is, if the ion channel opens or closes, resting membrane potential will change, and this change can be measured, which is important for transmitting the signal from one neuron to another. So change in the resting membrane potential is actually nerve impulse. So we can write over here, change in resting membrane potential is nerve impulse, or we also call it action potential. We also called it as action potential, nerve impulse or action potential. So this technique gives us idea about the activity of a specific ion channel on the surface, and it tells that how whole cell behave in opening and closing of the channel. This there is some issue in the system. Just give me a second. So yes. So this patch clamp technique gives us idea about two things. Patch clamp technique gives us idea about, first thing, specific ion channels, specific ion channel on surface, and second thing it tells is how whole cell behave in opening and closing of channel. How whole cells behave in opening and closing of channel. So these two things we are going to understand with the help of patch clamp technique. Now let us see that how the electrode and all, they are having their size and everything. We'll talk about that. We'll talk about this. So the electrode is in the size of 10 to 25 micron meter. The pipette is fire polished. Then it contains, the pipette contains electrolytes. These are some of the things. Then we'll be talking about patch clamp circuit. Patch clamp circuit. So we are going to draw this patch clamp circuit. This is the plasma membrane and these are the channels. This is one of the channel and this is the plasma membrane. Now the pipette will be attached on this. So it's a fire polished pipette. So it is going to be attached in this way. After that, in this pipette there will be certain solution so that it is mimicking the extracellular environment of the channel. After that, there will be an electrode which will be dipping in this solution in the pipette, and it is attached to an amplifier, because the amount of current would be very feeble, very small amount of current, because it is in the body. So the amount of current cannot be very high, which can be easily detected. So it will be connected to an amplifier, and this whole thing will be connected to a recorder. Whole thing would be connected to a recorder. There will be a recorder and resistor attached to this, and the another part of the another electrode would be connected to the ground. The another electrode would be connected to the ground, so that we can get a reference. So let us just label the things. This is the patch of cell membrane with ion channel, and this is like whole membrane going on like this. We haven't cut out the part from the cell. So this tells you about patch of cell membrane with ion channel. So talking a little bit about the technical issues over here. See, the high gain operational amplifier is connected to the circuit. This amplifier is connected to the circuit, so that the current flowing through the ion channel is measured as a voltage drop across the feedback resistor. This is the resistor. This is the feedback resistor, and the feedback resistor has got a resistance of 50 giga ohms, allowing very small current to be measured. So if we are saying very small current, so what range of current and what power of current can be measured? So you will be shocked to know that the current in the range of 10 raised to the power minus 12 amperes can be measured along this whole setup. So this is the patch clamp circuit. This is the patch of cell membrane with ion channel. This is the pipette, and in the pipette there is a an environment of solution which is mimicking the extracellular environment, and this will be the intracellular environment of the whole channel. This is how it is going to actually give it a normal behavior or a normal setup. Now we'll be talking about the technique, that how it is performed in the flowchart diagram form, and then we'll be talking about some types. So what is the technique? So in this technique, we take tissue or a slice of tissue, and this can be obtained from animal cell culture as well. So we take slice of tissue or animal cell culture, and this would be put under microscope, so that only that particular channel can be observed. So put under microscope, and this for whole thing we need just one single isolated cell. We need single isolated cell. A pipette will be used which will be very thin in diameter. A pipette with thin diameter, and diameter we have seen, thin diameter is used at the channel. So it will be inserted at a surface, and mouth of the pipette is smoothed out with the help of fire, and the pipette contains conducting particles, the ions which are actually present in the extracellular environment of the normal cell. So conducting particles, After that, there will be suction at the end of the pipette, and there will be an electrode. We have seen this in the diagram. So at the end, there will be suction. There will be suction and an electrode. And this will be attached with an amplifier. After amplifier, there will be a receptors to record the whole current, and on amplifier and all there will be a resistor, right? So if sodium flows out, membrane potential will change and the signals are going to change, and this is how we are going to get a recorded current, right? Now we'll be talking about configuration of patch clamp. Configuration of patch clamp. So first is on cell, second is inside out, third is whole cell, and fourth is outside cell. Outside cell. So on cell patch, how will it be? On cell. This is on cell patch. After that, inside out patch. This is inside out patch. Then whole cell clamp. This is whole cell clamp. Then outside out, outside out. This is outside out configuration of patch clamp. So these are some of the configurations of patch clamp. Now there are types of patch clamp that we'll be seeing. Types of patch clamp. First is perforated clamp, perforated patch, and second type is loose patch. Perforated patch and loose patch. So we'll be seeing a perforated patch. See, see, this is how a perforated patch is going to look like. This is our pipette. So these are the ion channels, and this is the glass micropipette, glass micropipette, and there will be solution in the pipette. This is the pipette solution, plus nystatin solution, This is the pipette solution, and this is the cell membrane, and this is the nystatin pore. This is nystatin pore. And this is the picture of perforated patch. This is the picture of perforated patch in a very, very simplified manner. So there are two types of patches, perforated patch and loose patch. So let us talk about patch clamp technique in isolated cardiac myocytes. Patch clamp technique in isolated cardiac myocytes. Cardiac myocytes. So, perfusion of a section of intact canine left ventricular myocardium was taken. A cannula has been placed into the left anterior descending coronary artery and clamps have been placed to occlude major coronary artery branches and they have been transected during the sectioning. So that is how we are going to get a single tissue from a canine. So perfusion of a section of intact canine, left ventricular myocardium, then a cannula. That thing when we get a drip, whenever we have to get a drip, we are fixed with the cannula. So a cannula has been placed into the left anterior descending coronary artery and clamps have been placed to occlude the major coronary artery branches and have been transected during the sectioning. So how the myocytes they are obtained in this case, because we would need single cells. So how will it be done? Isolation of myocytes. Isolation of myocytes. So how is this done? Intact heart section would be done. Then wedge preparation would be done. A small part would be taken out. So wedge preparation. Then perfusion and dissection would be performed. Perfusion and dissection would be performed. After that, epicardial preparation would be done. Epicardial preparation would be done. After that, batch dissection and filtration would be done. Batch digestion and filtration would be done. And like this, we are going to get isolated myocytes. Isolated myocytes, and which is the most important thing for performing the technique. And then this will be subjected into incubation buffer so that they do not lose their activity, and then it will be put under electrophysiological study, that is patch clamp technique. Electrophysiologic study will be subjected to electrophysiologic study. So isolation of myocytes we have seen for a male Vista rat. Male rat we would write. So intact heart was taken out by dissection. Then small wedges are prepared from that intact heart. Then perfusion and dissection was done and epicardial preparation was obtained, and they were subjected to batch digestion and filtration, and isolated alive myocytes were obtained. And then they were subjected to the procedure. So coming to the principle and procedure of patch clamp technique. Principle and procedure. So the generation of an action potential in the heart muscle cells will depend on the opening and closing of the ion selective channels in the plasma membrane, right? So first is generation of action potential. Generation of action potential will depend on opening and closing of ion selective channels ion selective channels in plasma membrane. First thing, then the patch clamp technique will enable the investigation of drug interaction with the ion channel, because it will be present in that pipette. So it will be very easy for us to to understand the drug interaction with the channel. So second is patch clamp technique enables the investigation of drug interaction with ion channel, and the isolated cells are ready for the experiment. Glass micropipette will be having a tip of about 1 micron meter and it will be placed onto the cell. So micropipette tip is in the size of 1 micron meter. So it will be placed on the cell. Then the pipette, it will be filled with either NaCl or KCl solution and it will be mounted on the micromanipulator, right? So pipette will be filled with NaCl or KCl. Then a chloride silver wire connects the pipette solution to the head of the electrical amplifier. We have drawn this diagram in the starting. So the electrode is of chlorided silver. Electrode chlorided silver wire and it is connected to the electronical amplifier. And the another electrode will be connected in, or it will be inserted into the ground, or it will be connected to the bath. If you are having any kind of doubt, which I am feeling, we'll be just going back to the diagram which we drew for this. See here, this is the silver chloride. See, this is the silver chloridated electrode and the second one, it is connected to the ground or it is connected with the path. So the two electrodes, right? This in this picture, the thing is quite clear. So the next thing, the next step, it is, I hope this is the sixth one, yeah. So next is second chloridated silver wire or the electrode inserted into bath and serves as ground electrode. So this is how the whole patch clamping is done in this way. After this, there will be the high input resistance which will enable the recording of the small electrical currents in the range of picosiemens, which are actually flowing through the channel performing the protein situated in the membrane patch. So high input as a resistance would be set up on this. Then the electrical current is driven by applying the electrical potential across. I'll just go to the picture so that the things would be clear. See over here, a high input resistance. This is the high input resistance which is actually put up, and this is of 50 giga ohms. This and which are flowing through the channel performing the protein situated in the membrane potential. Then the electrical current is driven by applying the electrical potential across the membrane patch or by establishing an appropriate chemical gradient here for the respective ion species. A gradient should be created so that there can be flow of the current. Now another question in this case is to study the interaction of drugs. So to investigate or to study interaction of drugs, with ion channels involved in the functioning of the heart cells, what all things we are going to need. We will need potassium ion, we'll need sodium ion, we'll need calcium ion, and we'll need chloride channels. So all these things would be needed. So different types of potassium channels exist in the cardiomyocytes. So just for one type of ion, there are different type of channels. So this is how the process of patch clamp technique is done, and it is used for various type of drug interaction and membrane potentials. So finally concluding the whole thing and talking about the patch clamp technique in a very concluding way, conclusions. So in conclusion, we'll be talking about application, we'll be talking about limitation and all the things. So it's a highly modified and successful technique, and development of this technique is being done for newer approaches to yield better, accurate and efficient information which is related to the drug discovery. So conclusion, very uh, important technique for drug discovery processes, for drug discovery processes, and it is a very successful technique as well. Now recent research, what it says about patch clamp technique is measurements are conducted in a multi-parametric manner in an integrated and automated microfluid chips. So various parameters can be applied to a single patch clamp technique. Micropipettes which were used in traditional patch clamp technique are nowadays replaced by nano machine patch clamps. So nano machine patch clamps are used these days with integrated microfluids, and the microfluids aids in rapid intracellular perfusion, improved optical measurements and rapid measurement of single cell dose response curve. So nano machine patch clamp, integrated microfluids which aids in rapid intracellular perfusion, rapid intracellular perfusion, then improved optical measurements, then rapid measurement of single dose response curve, single dose response curves. These all are the benefits of use of nano machine patch clamp which are integrated with the microfluids. Now talking about some of the applications, because we have seen for drug discovery, okay fine, we are talking about it since quite long, but what is the actual other applications, because just one application cannot do for everything, right? So it is used for evaluation of antirhythmic agents, evaluation of anti-rhythmic or antirhythmic agents. Then it can be used for kidney cells. This technique can be used for studying the kidney cells. Then it can be used for isolated ventricular myocytes, isolated ventricular myocytes obtained from guinea pigs to study a cardioselective inhibition of the ATP sensitive potassium channel. Then to identify multiple types of calcium channels, to identify multiple types of calcium channels, to measure the effect of potassium channel opens, how it opens, then it can be used in molecular biology. Then voltage clamp techniques for sodium channel, it can be used. Then measurement of cell membrane conductance, cell membrane, conductance. These all are some of the applications of patch clamp technique. Now anything cannot come without any kind of limitation. So we'll be talking about limitations as well. So limitations of patch clamp technique. So imparting skillful training, performance and recording during the whole channel is quite crucial. So imparting skillful training, performance and recording in and during a single channel recording is quite crucial. Then cost of the process is expensive because it needs sacrificing the whole animal of your, or we are going for animal tissue culture or animal cell culture, then too it is quite expensive. Then it is time consuming. Then number of sample required is more at times. Number of samples should be high. Then chance of membrane distortion is always there whenever we are putting up the pipette. So the chances of membrane distortion is high. So chances of membrane distortion is more. These are some of the limitations of patch clamp technique. Now finally, evaluation of the technique. So we'll be doing it here. Evaluation of the technique. So concentration response curve of drugs which either inhibit or activate the ion channel can be recorded either on a single channel level or by measuring the whole cell current. So these are some of the evaluationary thing that what we have to do. We are going for a single channel or we are going for a single single channel or single cell. So there are chances of different recordings if we are going for a single cell or if we are going for a single channel. So there are chances to be having different recordings. So this we have completed in a well systematic manner. We'll just do a very quick revision. So we were studying patch clamp technique, and it's an electrobiological technique used for measuring the electron or membrane potential for various neurobiological issues. It is used. Then it allows a study of single or multiple ion channels in the cell in a single go or in various different goes. It was developed by the two scientists, that is Sackman and Neher, and this for this they received a Nobel for their development of the technique in the year 1991. Then there are various historical developments since Jain did his study till Sackman and Neher. There are many changes which were done in this, and finally they came up with patch clamp technique. So what is the need of patch clamp technique? It provides low noise recording of the current of the cell because the cell has a potential. So to study this potential and how efficiently it transfers the ion and how sufficiently and rhythmatically it exchanges the things, so that can be very much studied by patch clamp technique. It provides access to the inside of the cells, so we can insert electrode into the cells and we can change the intracellular fluid, and by this it is creating a seal which is impermeable to ion flow and high electrical resistance. So it measures the current through the ion channel and versus voltage, time and temperature. The basic principle we have seen. Then we have seen that what this patch clamp technique tells us about. Then we have seen the whole setup of patch clamp circuit. After that we have seen the technique, and then we have seen the configuration of patch clamp in 4 different ways. Then types of patch clamps we have seen. Then we have seen how to isolate the myocyte. Then what is the principle and procedure behind the whole process. Then in conclusion we have seen that what all are its benefits and what all are its applications, and then in application we have seen where all places this can be used and some of the limitations which are associated with patch clamp technique that we have seen. And in evaluation we have seen that we can study a single channel or whole cell current or whole cell current. So this was the evaluation. So like this, we have completed this topic in almost a very systematic and a precise manner, so that you can attempt the questions in the examination. And you're watching this video on the channel Let's Crack CSIR UGC NET, and we are doing a crash course for GATE 2021. We'll be meeting on Monday again with another interesting topic of GATE syllabus. And as we say, let's crack CSIR UGC NET, and hit the bell icon for getting all the notification regarding the upcoming videos. Use my referral code FB01 for getting the plus subscription, for getting an additional 10% discount. Use my referral code FB01. And for special classes as well, use my referral code, that is FB01. And if you haven't subscribed our channel, subscribe it. And if you haven't liked our video yet, give it a thumbs up and like the video. It really motivates the educators like us. And lastly, a great thank you to one and all for your patient listening, and hope to see you in the class of Monday. And we'll be doing some another interesting topic in the morning session and evening session. Morning we'll be talking about some topic of CSIR syllabus, and in evening we'll be talking about GATE syllabus. So this is it for now, and I have made a video on community ecology part 2 as well. So soon we'll be premiering the video of community ecology part 2. In that we have seen the vertical gradient of the water community. So just go there and watch the video and try to study well. So thank you very much. Have a very good day and be happy. Keep on studying.